Hi all, welcome to my channel. So today we'll talk about some of the pigments which the bacteria produces. This is one of the most commonly asked question for microbiology students. So for human pathogenic bacteria, we'll try to remember it with a mnemonic. So the mnemonic would be some microbes can bring stunning colors, making plates shine forever. So let's get inside and see one by one. So the first bacteria is our Staphylococcus aureus, which produces a beautiful golden yellow pigment. So this is called as a staphyloxanthin, which is a carotenoid pigment. It is lipid soluble and it is stored in the cell membrane. So this basically acts as an antioxidant. So what happens? So whenever there is a reactive oxygen species being produced by the host, this particular pigment helps the bacteria nullify the effect of that particular reactive oxygen species. So this pigment production is controlled by a few operons, that is your CRTM and CRTN operons, and this is one pigment which is beautifully linked to the virulence factors of Staphylococcus aureus. So next you have the Micrococcus. The next member is our Micrococcus species. This particular bacteria has a very bright lemon yellow kind of a pigment, which again a carotenoid pigment and is a lipid soluble pigment. So this pigment, like your staphylococcus, also incorporated in the bacterial cell membrane. Now it has antioxidant properties. Here the antioxidant helps the bacteria. Since this bacteria is a part of the normal skin flora, this particular pigment helps the bacteria resist the UV radiation. As such, Micrococcus does not have a role in causing infections except in immunocompromised hosts. Otherwise, it does not have any virulence properties attached to the Micrococcus species of pigment. Next, we have the Corynebacterium diphtheriae. As such, this is not a bacteria which produces a pigment. A few strains which will produce black colonies on the telluride agar, which does not have a major role. It only helps in the purpose of diagnosis. Now we will talk about the Bacillus subtilis group, in which a few strains can produce a melanin-like pigment. It's a dark polymerized compound which is derived from the metabolism of tyrosine and catechol. The pigment is non-diffusible, it acts as an antioxidant and protects the cells from the UV rays and environmental stresses. Generally, this is not considered as a virulence factor. Now we have the Streptomyces species. They have some secondary metabolites, including pigmented antibiotics. There are a variety of colors: blue, red, gray, white, and they are generally intracellular and are diffusible. They protect the bacteria against the other soil microbes, and some of them can act as antioxidants. Now, the pigment production is most important for antibiotic discovery. That is, some of the antibiotics we know tetracycline, streptomycin, actinomycin, all those things are from these particular species of the Streptomyces bacteria. Next, we have the Chromobacterium violaceum, which is basically produce a very bright violet purple pigment, which is an indole-derived compound. It's a diffusible pigment. It has antimicrobial, antioxidant, and cytotoxic properties. It helps this bacterium survive in the soil and aquatic environments, and this will help the bacteria survive. And more than the virulence properties, it is for the surveillance or survival of the bacterium. As we know, we have the Mycobacterium species, the non-tuberculous Mycobacterium. So again, the carotenoid pigments ranging from yellow to orange. So you have the photochromogens, which produce pigments only on exposure to light. You have the scotochromogens, which can produce pigments both in light and dark. You have the non-chromogens, which do not have much of a pigment, and this particular pigments help the bacterium protect itself against the oxidative damages and the UV stresses. As we all know, the famous Pseudomonas group, you have different pigments. You have the pyocyanin, which is a blue-green phenazine compound. You have the pyoverdin, which is a yellow-green fluorescent compound. You have the pyomelanin, which is brown-black, and you have the pyorubin, which is a red-brown compound, which is relatively uncommon. Now, these are generally useful in virulence. The pyocyanin generates some reactive oxygen species, damages the host cells. You have the pyoverdin, which is an iron-related compound essential for bacterial growth in areas where the iron compound is minimal, and other pigments which will help in resisting the oxidative stresses and for the persistence of the bacteria. Next, we have the Serratia group, the Serratia macerans, which is a red compound, which is your producing, which is a water insoluble compound. These colonies, they are bright red in room temperature. Sometimes you don't see this pigment at 37 degrees. You leave it, you leave your plate outside for a few hours in the room temperature. They will develop this pigment, and helps this bacterium compete with other microbes. Not much of a role in human virulence, and it can act as a opportunistic infection or nosocomial infections in patients. Now we have the Flavobacterium species, which again produces carotenoid pigments, xanthophylls, into yellow to orange coloration. The pigment is non-diffusible and it acts as antioxidant to protect against oxidative stresses. As such, there is no role in human virulence. Apart from the bacteria, what we have studied till now, we have some gram positives like the Actinomyces and Nocardia group, the Rhodococcus brevibacterium among the gram positives. Among the gram negatives, very rarely some of the Klebsiella pneumoniae strains and Vibrio cholerae strains may produce a pigment. And you have the Altromonas, Schwanella, and Porphyromonas group which produces the pigment. So, among the human pathogens, we'll try to remember them with a mnemonic. Some microbes can bring stunning colors, making plates shine forever. Hopefully, you like the video. Please drop in your comments or suggestions in the comments box. Thank you.